Bunsen burners provide a sterile zone for proper aseptic technique and sterilized inoculating loops. Before lighting the Bunsen burner, check the flint lighter to ensure that it has flint. The amount of gas entering the Bunsen burner can be adjusted by moving the valve on the bench. Adjust the gas by dialing the screw on the Bunsen burner. The amount of oxygen in the Bunsen burner can be adjusted by turning the dial above the gas screw. In the lab, working with a blue cone is essential for proper sterilization. To sterilize the loop, place the loop in the region between the inner blue cone and the outer part of the flame at an angle. The loop will glow red hot. Move the wire through the flame and slowly draw the loop out. Do not touch the loop, allow it to cool for several seconds instead. Do not reach over the flame to take something from the riser. Carefully move the burner from its base to the side before lifting your arm. The incinerator is used to sterilize the loop when you are working with biohazard safety level 2 pathogens. Turn on the incinerator as soon as you enter the lab to allow it to warm up. To sterilize the loop, place the loop inside the heating element, but ensure that it does not scrape the sides. Heat it until it is red hot, then remove it from the incinerator. Do not touch it, and instead allow it to cool for several seconds. The ethanol jar is used to sterilize glass spreading rods, which resemble hockey sticks. Dip the hockey stick inside the ethanol, then pass it through the flame and allow the ethanol to burn off. Hold the hockey stick down at all times and do not leave the hockey stick inside the flame. If a flaming drop of ethanol falls off the hockey stick and onto the bench top, let it burn off. If the jar of ethanol is lit on fire, cover the jar with its lid or your lab book. The flame will burn out quickly. It is important to note that the ethanol sterilizes the hockey stick rather than the flame. The flame is only used to burn off the ethanol. It is essential to dispose of equipment properly and handle broken glassware and spills safely. Once you have finished using a pipette, remove the bulb and place the pipette in the pipette bin with its tip facing downwards. The discard beaker is used for Pasteur pipettes, swabs, and pipette men tips. When you remove your gloves, remove the second glove by pulling from the inside of it with your exposed hand to avoid contamination. Place the gloves inside the glove discard bin. When you are finished using cultures, place them upright in the test tube racks in the side bench discard bins. Do not lay them down inside the bins. If liquid culture spills on the bench top, soak up the liquid with paper towel, then spray the area with bench cleaner. After applying the bench cleaner, you must wait for a few minutes to ensure that disinfection is complete before cleaning up the paper towels and placing them all into the red biohazard bag. As for broken glass spills, ensure that all of the glass is swept up with a disposable dustpan and broom, first before wiping up excess liquid with paper towels and bench cleaner. The dustpan, broom, and all of the paper towels belong in the red biohazard bag, which is placed in the discard bin at the side bench to be autoclaved and then discarded.